All right, guys, welcome to our March Tunnel Talk session. We'll go ahead and get started here. So today we're going to be looking at some product efficacy against botrytis. So just a few upcoming events that I've sort of earmarked may be of interest. We have our April session of the webinar, which is going to be looking at pollination. And then in May, we're going to take a look at biological spray programs. Those will be specific to tomato and strawberry, but we're also going to go through Perennia's new sort of spray guide format. We've moved away from our Word document multi-page system to something a bit more interactive, still printable. Yeah, it's pretty cool. Our berry specialist, Sunny Murray, is going to be doing his berry primer for the season on March 21st. So that is always a good one to jump onto. Tons of speakers on that one. Horticulture Nova Scotia is going to be doing an irrigation session on April 9th at Spur Brothers. This is going to be more geared towards field production and looking at drainage and various equipment sort of surrounding those topics as opposed to a greenhouse session, but definitely worth checking out if you're interested in that. Perennia is going to be hosting a series of introductory sprayer training sessions, just kind of going over the basics, making sure that everyone knows everything there is to know if we're spraying or new to spraying or playing with new equipment, that sort of thing. So those will be in April. Stay tuned for our to our newsletter or to the vegetable blog, and that will have notes about when those dates are. Our tech team is also going to be hosting some weather station maintenance workshops May through July. I think the goal for those as well is to cover most of Nova Scotia. And again, that's covered on perennia.ca/learning. Also, I forgot to mention there are going to be pesticide points today. So stay tuned for the link at the end of the presentation. Yeah, get you some points. And just a quick announcement about off-CAF funding. So we have officially been accepted to deliver a third year of the program that we've been implementing the last 2 years. We're still looking to build adoption of best management practices surrounding split nitrogen application, cover cropping, and rotational grazing. This is pretty specific to new adopters and new land, but it could be worth a call to our technical lead, Georgia Lewis, if you're looking to improve implementation of those practices on your farm. Intake closes April 12th, so we've got a pretty short turnaround period for that one, but yeah, a lot of great opportunities there. So with that, I'm going to introduce our feature speaker for today. Annissa is a plant pathologist with over 18 years experience in crop protection and crop enhancement research and development. She's got a background in microbiology, plant biology, and horticulture. She has a master's and PhD in plant pathology from Penn State, and where she studied biocontrol strategies for managing weeds and diseases of tree fruit. Prior to her current role as manager, she worked as an assistant professor at the University of New Hampshire and as a research scientist at Vineland Research and Innovation Center. So with that, Annissa, I'm going to turn it around to you.
Great. Okay, good morning, everybody. Let me share my screen. We'll get started. Okay, how does that look?
Nailed it. 2 for 2.
All right. So, yeah, so I'm going to talk about specifically a lot of the work that I've done recently with seafood byproduct chitosan and integration with biologicals for managing gray mold. Mostly this will be in floriculture, but there's applicability across all different kinds of crops. So this is work that I did at the University of New Hampshire, and pretty much a lot of the work I'm going to show was done by Liza DeGenring, who was a graduate student but is now a postdoc at the University of Minnesota. So Talia introduced a little bit of my background. I thought I'd just tell you a little bit more about who I am. So as she mentioned, I'm a plant pathologist, and most of my training was in weed biocontrol and tree fruit biocontrol. But in the last 10, 15 years, most of my work has been on biologicals and biostimulants in greenhouse production, both vegetable and floriculture. And I also do a lot with sanitation and soilless substrate. So I really got into greenhouse production and a little bit into high tunnels when I was a research scientist at the Vineland Research and Innovation Center in Southern Ontario. And that's actually where Talia and I first met. So I worked on disinfectants, rose breeding, root rot control and greenhouse production, and water treatment methods for removal of pathogens. Then I spent about 6 years at the University of New Hampshire, and that's where I started getting into botrytis and the compound chitosan for disease management, but also been working with wood fiber substrates. In September, I started at Mycorrhizal Applications as the product development manager, and I oversee our mycorrhizae products as well as our biopesticides for disease control. So we'll start off today kind of setting the stage with IPM, or integrated pest management, which is this tool or decision-making process where we combine lots of different tools that maybe individually are not as effective, but collectively are effective. And the goal here is to minimize and manage risk that's associated with pests and pathogens, but we do this in a way that also considers economic risks, health risks, and environmental risks. And a lot of what we're doing by integrating some biological tools are to minimize pesticide resistance so that our chemical controls remain effective and that we don't lose them. And so we do have some valuable chemical tools, and we want to make sure that those are around for the long haul. And so part of that involves integrating biological tools. And these are tools in what I call our IPM toolbox. And we have our hammers and we have our nails. And so we have certain things that are our heavy hitters that we can rely on to clean up a problem or do a really good job at preventing. And so we have our chemical pesticides, but then we also have our biological pesticides, and these can take many forms. So we have some biologicals that are called biochemical pesticides, and that they don't contain a living organism. So usually these are the compounds that microbes produce that have antimicrobial activity. There are also plant extracts that have antimicrobial activity. Then we have our microbial biopesticides that contain living organisms, such as fungi, yeast, bacteria. We also have biostimulants and biofertilizers, which are becoming more and more popular. And these, while they don't claim to have biopesticide activity, they're really to help plants uptake water and nutrients, essentially by promoting plant health. It helps plants then defend themselves against diseases and pests. And so we often see this effect in reducing diseases just by simply enhancing the health of the plant overall. So we use these tools in part of our IPM program, where we produce or we build a solid foundation of cultural controls using plant resistance when possible, sanitation, understanding the biology, forecasting, and monitoring for outbreaks, and then providing or applying our tools. So we try to build up on prevention where we can minimize the need for intervention. So as I mentioned, I'm going to spend some time talking about biopesticides today, and these can take many forms. So they can range from metabolites that microbes produce, the microbes themselves, plant extracts. So regalia would be an example of a plant extract. And then we also have natural compounds. And biopesticides can be a useful tool, but they're rarely effective on their own. They are best when combined with other strategies, and they can be challenging, or they can be frustrating sometimes because of the variable efficacy, where they may work really well in one year and not as well another year, or they may vary from field to field or crop to crop. And one thing that is an area of research that I've been working in a lot lately is, what can we combine with biopesticides to make them more effective? And one option is combining biopesticides with natural products or biologically active agents that can improve disease suppression. So this is taking biocontrol microbes and combining them with these compounds. Some of these compounds include chitin and chitosan, which I'll talk about today. And then there are some other naturally occurring compounds. And often what we see are these enhanced plant health and disease control. So we see a synergism in that when we put the two together, it's more effective than either of them alone. So I'm going to spend some time introducing you to chitosan, which is derived from chitin. Chitin is one of the most abundant polymers on Earth, and it is then we can derive chitosan from the deacetylation of chitin, and this is a process that we use through the addition of an acid. And so the term chitosan is not necessarily a one single compound, but there's sort of a group of polymers that chitosan describes. And chitosan has historically been a niche biopolymer in that it was pretty expensive to produce, it was inconvenient to use, and it's not soluble in water, so it has to be dissolved in acid. And so that provided some challenges in creating a product. And so this traditional process by which chitin is extracted from crab shells and crustaceans and then converted to chitosan was created a lot of waste, and that was part of why it was so expensive. And so there really needed to be a substantial investment by companies into R&D and creation of specialized equipment to bring those costs down. And this has happened more recently, making this more available, and we're starting to see products become available because the costs have come down. So chitosan is actually used in many industrial applications. It's used in pharmaceuticals, it's used in water treatment. So it's an eco-friendly flocculant for water clarification because it binds a variety of organic and inorganic particulates. It's also used in cosmetics, in textiles, food preservation. When I first heard of it, it was used for post-harvest treatment of perishable fruits to prevent post-harvest decay, but it's becoming more popular in agriculture as a crop protection product. And the reason for that is that chitosan has many plant health benefits. So chitosan and even chitin itself, which is primarily found in insect and crustacean exoskeletons, but it's also found in the cell walls of fungi, is known to induce plant defense responses. So plants respond to chitin and sort of boost their immune system, similar to some of the resistance inducers that we see on the market, like Actigard or Regalia. And so the plant is better able to defend itself and more tolerant to stress in general when the immune system has been activated. So we also know that chitosan has antimicrobial activity itself. It's been shown to inhibit growth of many fungi and also bacteria. And then we also see that chitosan stimulates the production of antimicrobial activities by our beneficial microbes. So essentially, many microbes, especially bacteria and some fungi, can actually use chitosan as a food source. And in order for them, so when microbes are utilizing a food source, they produce enzymes similar to what the enzymes we produce in our gut to be able to break down that material as a food source. Those enzymes that they use to essentially eat chitosan have antimicrobial properties. So when these microbes are in the presence of chitosan, they produce these antimicrobial enzymes in order for them to digest chitosan. So you get this benefit of the production of these enzymes. And we also see this benefit where chitosan is a food source for biocontrol microbes, which can be beneficial when we think about the leaf surface and control of foliar diseases, because one of the things that biopesticides struggle with when applied to the leaf surface is a pretty inhospitable environment for microbes. There's not a lot of food on the leaf surface. It is exposed to fluxes in temperature, humidity, you have UV radiation exposure. And so by packaging microbes with a food source, it gives them that boost so that they can survive on the leaf surface and do what we want them to do. So there's been a ton of research by university researchers and more recently in the private sector, showing that chitosan can reduce disease, both as a foliar spray and in the post-harvest realm for extending shelf life in many crops. And where I first found about it, and really where I think it first made headwind, was its ability to reduce post-harvest rots and extend shelf life. So there are lots of examples, especially on strawberries and apples, showing that chitosan-coated fruit develop much less rot in storage. And this is showing botrytis on strawberries and blue mold on apples. So I started working with chitosan back in 2010 when I was a graduate student at Penn State University. And I was reading all this literature about the shelf life properties of chitosan, and I started asking these questions about, well, what if we combined chitosan with biologicals? Would that reduce the variability with biologicals? And I had some really good results where I found that if I applied chitosan or chitosan plus a biocontrol, I was able to significantly reduce fruit rots caused by polytotricum, which is bitter rot, and also penicillium, which causes blue mold. But I did this research and I found some really great results, but then I didn't really work with it again until 2020 when I was at UNH. And the reason why there was a 10-year gap from my interest in this topic was there were no products available. So I had to order chitosan from a research chemical supplier, and I had to make it myself. So I would get the dry flakes, I would have to dissolve it in acid and make the solutions myself. And so I was wanting to do research on things that would be directly applicable to growers. And I knew that this had great potential, but it wasn't something that a grower could go out and use because it wasn't available. And at the time, this was still in that realm of it was too expensive, and the process of making it into a product was difficult. But now it's probably around 2019, 2020, we start to see that companies are beginning to utilize waste from the seafood industry as a source of chitosan, and new technologies have been developed for extracting chitin from crustacean shells and then producing chitosan. So those costs have come down. And so we're starting to see products on the market. And so at UNH, I resurrected all the old research from 10 years ago to see how these products performed as crop protection products. So two products that we worked with were from two different companies. So one company is called Tidal Vision. They're based out of Washington State in the US, and their product, Tidal Grow Agriscience, and they've since modified their formulations, in part because of the research that we've done with them to provide them some direction of how they could modify their formulation. And then Botrytis is a company that's based out of New Zealand, and their product is Armorzin. And you may also be familiar with Botrytis. That's also a Botrytis product that is distributed by BioWorks. And so in the U.S., the U.S. Food and Drug Association has labeled chitosan as non-toxic, biodegradable, and biocompatible. Some of these companies have been pursuing organic status, but as of now, it is not organic, because if your source of chitosan is from crustacean cells, that process of converting chitin to chitosan doesn't happen naturally. And so it's considered an artificial process and thus not an organic compound. But you can also get chitosan from fungi. And so that source may open the doors for organic, but right now it's not organic. So some of the factors that influence efficacy of chitosan in agriculture are listed here. So one of the factors is the molecular weight. So I'm starting over here on the top left. And so this essentially depends on how many monomeric units are in the biopolymer. So sort of in how large it is in a sense. And the molecular weight influences the solubility. So the larger the molecular weight, the less soluble it is in water. And chitosan is categorized in 3 different ways. So based on molecular weight, it's either listed as low, medium, or high. And there's been debate among scientists which is better for disease control. Another factor, moving over to the top right, is the source material. So chitosan is most commonly obtained from crustacean shells, so crab, shrimp, and that's why it's seen as sort of ecologically beneficial, because we're taking a waste product from the seafood industry and turning it into a crop protection product. But you can also obtain fungi, chitosan from fungi and from insect cuticles. So it's thought that perhaps the fungal source may be beneficial, and that it also sort of meets the vegan market, and that it's not from an animal. Next is the method of extraction. So we have to extract chitin from crustacean shells, and there's various different ways to do that, and then to process the chitosan or the chitin into chitosan. So those processes can vary in cost and then result in a different end product. Then once we have our chitosan, we are putting that into a formulation. So we're adding inner ingredients that serve as a carrier for the active ingredient that can help with stability, storage, and also help the product sort of there's spreader stickers that may be added to the formulation. So different products may vary in those inner ingredients that can affect efficacy. We also think about compatibility with other tools. So that's a question that I've been asking, is how does chitosan work with some of the other management practices that we would use in our production systems? And one of the questions I've asked is it compatible with biopesticides? Another is the dose or the concentration. We know that the higher the concentration of chitosan is in a product, the more effective it is for reducing disease. But we also know that high concentrations can cause phytotoxicity, and those phytotoxicity levels vary by the crop, which I'll get into in a minute. Because chitosan is not soluble in water, it has to be dissolved in acid. And so the solutions tend to be low in pH. They can be anywhere from 3 to 5. And so part of the phytotoxicity issue is related to the low pH. And then finally, tied to molecular weight is this degree of decetylation. And this determines the molecular weight. And so you'll see varying degrees of decetylation. Typically, it's around 70% that are used in industrial products. So all of these things influence how the product works. And some of these were questions that we started asking to be able to provide recommendations to growers for best use practices and to inform some of the companies in their R&D as they're making adjustments to their product and to their formulation. So our goal was to identify benefits of chitosan. Does this work? And what capacity could we use it, and some of the best use practices. So we evaluated the antimicrobial activity against several plant pathogens in the lab. We looked at the different molecular weights. So that was one thing I said that could influence the efficacy, to see which one, did it matter, and if one was better than the other. We looked at compatibility between these commercially available chitosan products and commercially available biologicals, and then evaluated combinations of chitosan and biopesticides for suppression of botrytis. So botrytis was our model that we used to ask a lot of these questions. So a lot of the work I'm going to show you is looking at chitosan in greenhouse ornamentals. So petunia was one of our model crops that we used for looking at suppression of botrytis. So as I mentioned, chitosan is known to have direct antimicrobial activity, but we wanted to see how it stacked up against botrytis. So what we would do is a Petri dish assay in the lab, where we would amend the media with chitosan and compare that to media that did not have chitosan. And then we would add our botrytis and measure the growth over time. And so we evaluated reagent grade chitosan that we would make ourselves, and we looked at high, medium, and low molecular weight. And then we also evaluated the commercially available products, Tidal Grow and Armazen, prepared at a range of concentrations. And so we did confirm the antimicrobial activity and suppression of growth of botrytis. So these Petri dishes, it's a little faint, but on the top, you can see the Petri dish is amended with water, so no chitosan. And you can see the botrytis growing out from the center plug, whereas in the Petri dishes on the bottom, this is Tidal Grow prepared at 0.5% chitosan, and there's almost no growth beyond this little plug that we put on the plate. So it's showing inhibition, and we saw that across pretty much any of the treatments that we saw. So then we wanted to look at chitosan rates on leaves that were effective at suppressing disease but didn't cause phytotoxicity. So on the right, this is showing petunia plants that have been treated with chitosan compared to water. And then we also compared it to acetic acid, because chitosan, as I mentioned, is dissolved in acid, and oftentimes that acid is acetic acid. So it's a chitosan acetic acid solution. So we wanted to know, if there is phytotoxicity, is it the chitosan or is it really just the acetic acid? And so we found with petunia, this is 4 days after we applied the chitosan, that you did start to see phytotoxicity at concentrations above 0.5%. We've done this with other plants as well. We've done a lot of work with apples. You can apply much higher concentrations of chitosan to apple fruit or even apple leaves, because especially as they mature, they have a waxier, thicker cuticle that can handle the higher concentrations. So our recommendation is, and to the companies producing chitosan, is to pay attention to the recommended rates based on the crop, because the rates will vary, because what you could use on one crop might cause phytotoxicity on another. So again, we looked at those different molecular weights, and on the left, these are petunia leaves that have been inoculated with botrytis with a low, medium, and high molecular weight chitosan, ranging from 0%, so no chitosan, up to 0.5%. And you can see that with some of the 0.5%, it looks like you're starting to see a lesion develop, and we think that that's partially due to phytotoxicity causing damage to the leaf. And because botrytis is a necrotroph, meaning it feeds on dead tissue, that's why we started to see botrytis in the 0.5%. So It wasn't necessarily that 0.5% wasn't effective, is that it was damaging the leaf, causing an opportunity for botrytis. But we did see some differences in the low molecular weight around the 0.2 to 0.4%, where we had a really good suppression of the development of the symptoms on the petunia leaves. We also looked at flowers. And so we looked at phytotoxicity and disease reduction on flowers. So we had to go a little bit lower on the flowers because they're a much more sensitive tissue than leaves. So again, this was on petunia. So we sprayed the petunia plants with the different treatments of either Armour-Zen or Tidal Grow or water. And then we inoculated those flowers with botrytis, and then we evaluated. So on the left is showing phytotoxicity caused by chitosan. So our water control here on the left is our not chitosan treated flower, where you can see what it would look like normally. And we did start to see phytotoxicity at the higher rates. So here you see with the Armour-Zen at 0.3%. And the Tidal Grow at 0.3%, you're starting to see some phytotoxic effect. We combined, in later experiments, we combined chitosan with capsyl, and we found that that did reduce the phytotoxic effects. And we think it's because it just helps spread the chitosan, because without capsule, the chitosan tends to bead up, and then you'll get these lesions in the parts where the chitosan was beading up. So on the right, this is showing our gray mold results. So we tried the chitosan at either 0.2 or 0.3%. And then we looked at, so Tidal Grow 4%, that it's a 4% solution, but it was prepared at either 0.2 or 0.3%. And Tidal Grow makes a low molecular weight product, and they also make a high molecular weight product. And then Armour-Zen, and then our water control. So here you can see with the water control, the flower is almost completely dead and taken over by botrytis. But we saw some really great results with the Tidal Grow low molecular weight, with low phytotoxicity and low botrytis symptoms, given that we kind of really killed the flower in the control. So this was really exciting to see, because it's quite difficult to control botrytis on flowers. It's much easier on leaves than it is on flowers, particularly for biologicals. So we also did this on leaves. So we would spray our plants with chitosan, and then we would inoculate the plants, either a whole plant, or we would detach the leaves with botrytis, and then measure growth over time to see if the treatments reduce the symptom development. And so here on the right, you can see the actual lesion size, and then I like the pictures of the leaves because it really tells the story. So from the left, we have our water control. You can see the botrytis lesion in the center of the leaf. We also combined the products with capsule, so we just had our capsule control. And we really saw the best results, again, with the low molecular weight. This is the reagent grade from the left, but then we also saw some great results with the Armour-Zen at 0.4% on the leaves. So we saw in some cases the Tidal Grow worked really well on the flowers, and the Armour-Zen worked really well on the leaves. But in most cases, the Armour-Zen and the Tidal Grow or the reagent grade chitosan significantly reduced, if not eliminated, development of botrytis symptoms on the leaves. So the next thing we wanted to do was, okay, chitosan seems to have great promise. These products have great promise for suppression of botrytis on leaves and flowers. What about compatibility with biopesticides? Because if it is antimicrobial, is it going to kill our biologicals, and should we not mix them together? So in order to answer that, we sort of simulated a tank mix. So this top is showing a very small scale simulated tank mix where we would combine chitosan and a biopesticide, or we would have the biopesticide alone. And then we would plate it to see the growth of the biological on this nutrient media. So we were checking for viability. And we found that there was no effect of chitosan on biopesticide viability. And we looked at, and I'll show some of the products in the next slide, both fungal and bacterial-based biopesticides. So far it looks like that, yes, they are compatible, and the chitosan does not harm the biological when applied as a tank mix. Then we also looked to see if there were any synergisms. Does chitosan enhance the activity of biologicals? And so we did this in a Petri dish assay where we would have our Botrytis on the Petri dish, and then we would have our biocontrol agent, and we would look for reduction in growth of Botrytis. And we found that when we combined chitosan and the biologicals, we saw a greater reduction of Botrytis growth in the dish than with the biopesticide alone. And we saw the best result is if we had exposed this biocontrol agent to chitosan first, and then put the biocontrol agent on the Petri dish with Botrytis. So we called that our chitosan activated biopesticide. That was where we saw the greatest suppression. So it appears that there may be a synergism here where the combination is better than either alone. So this is just kind of the data version of what I was explaining. So we looked at root shield WP, we looked at Botrytis, we looked at cease, that's a bacillus-based product, and then the BW165E is also a bacillus, a Bioworks product. It's a bacillus-based product. And this is just showing that when the biocontrol agent is grown by itself in water, was essentially no different in terms of its viability than when we mixed it with any of the chitosan treatments, so Tidal Grow or Armour-Zen at 2 different concentrations. And in some cases, you actually saw, like for cease, for example, and the BW165E, it almost looked like there were more cells of the bacteria in the presence of chitosan than not. And it might have been that the bacteria were using the chitosan as a food source, and it actually stimulated their growth. So we looked at the combination of biopesticides with chitosan on plants, and we've done this again with our friend Petunia on leaves and flowers. And this is just a snapshot of some of what we saw. So we looked in this case at 3 different biocontrols compared to water, so no biopesticides. We looked at cease again, which is the bacterial-based biopesticide, Howler, which is also bacterial-based, and then Botrytis, which is fungal. And then we looked at Tidal Grow at 4%, and then the bottom row is water, so no chitosan. And it looks like when we combined, so this bottom right-hand leaf where we have the water, that's our control. So that's Botrytis with no biological and no chitosan. So you can see the extent of the damage caused by Botrytis on that flower. And it looks like when we combined Botrytis or Howler with Tidal Grow, we had the best results. And this was the 4% product applied to the flowers. And so you're still seeing some damage because we had a pretty high level of Botrytis. And so it would be interesting to see if we had lower levels of Botrytis, so to mimic sort of low to moderate disease pressure, if we could completely eliminate symptoms on the flowers. So this was exciting to see that, yes, it looks like for many biologicals, chitosan is compatible and they can be mixed together, and that when you apply them together, it enhances the efficacy of biopesticides. I'm not going to talk about it a lot, but we also have done a ton of work in tree fruits, so looking at chitosan in the orchard. And so we've looked at this for control of foliar diseases, apple scab, and also we looked at fire blight, and then we've looked at post-harvest control. And I have just a little bit of information to share for you today, but if you're interested, we've got a lot more on all the research we did in tree fruits. But we did sort of the same questions of, is chitosan effective, and can chitosan be combined with biologicals? So for example, we found that if we did a pre-harvest application, so applying the chitosan, and this was with Serenade, so this is a bacillus product from Bayer, applied to the trees, that actually reduced infections that developed in storage. So these are bitter rot infections. And we compared that to a grower standard program of synthetic fungicides. And our chitosan plus biopesticides was as effective as the grower standard synthetic program, which was really exciting to see, that it looked like the chitosan plus biopesticide combination was better than either chitosan or biopesticide alone, and similar in efficacy to the standard program. And we have done a ton of work, again, where we saw similar results with foliar diseases and other post-harvest diseases. But I wanted to highlight this one because this is where we saw the biggest benefit. So in terms of the use of chitosan in crop protection, I think this is still an emerging area, but I expect to see growth. What we know is that the chitosan efficacy can vary based on the source material, can vary based on the molecular weight, manufacturing conditions, and formulation. We know that higher concentrations lead to increased efficacy, but that comes with the caveat that chitosan can cause phytotoxicity, but the rates will vary based on the plant in the tissue. So you can't go as high in your concentration on flowers, for example, as you can on leaves. And it appears that it is compatible with biopesticides, and there's potential for synergism. And in addition to crop protection benefits, there's also been a lot of research showing that you can apply it to the soil as well. Either these formulated products, I've known growers that have taken crab shells and ground them up and incorporated it into their soil, where we see benefits in enhanced plant growth. We see induction of the plant immune system. We've seen increased seed germination, improvement of overall plant quality. So either this could be done to improve transplants or into the soil to provide enhanced tolerance to stress. So the two products that we tested, they are registered in the United States. I couldn't find data about registration in Canada. I know that Botrytis, which is the supplier of Armorzan, has been pursuing registration in Canada. I do know the Botrytis stop is coming and will be provided by Bioworks, but I am not sure about the tidal vision where they were at. I know they got stalled a little because they were trying to see if they could get organic certification. But there are other companies that are interested in adding a chitosan product to their product lineup. And so hopefully this information provided today just gives you a little bit of insight that if you were to see this or to play around with some of these products, that there does seem to be a lot of potential here. So this work, most of it was funded by the American Floral Endowment, but we also had some funding from the Northeast Sustainable Agriculture Research and Education Fund for our tree fruit work. And we collaborated with folks at Penn State for the tree fruit work, and also Ryan Dixon at the University of Arkansas with the ornamental work. So we have some scientific publications, but we also got some online reports. So the American Floral Endowment posts their newsletter. So this is a little less technical. So I have these two newsletters that are on the AFE website, which summarizes a lot of the work that we've done in Botrytis on floricultural crops. So if you wanted to visit those, or even just go to the American Floral Endowment website, you can find their newsletters. And they have a thrips and Botrytis newsletter that also contains a lot of great information of research done other projects with botrytis. So they have some other work done out of Clemson with biologicals for botrytis, and our work with biologicals and chitosan on botrytis is up there as well. So that's what I had for today, and I'd be happy to open it up for any questions that you may have.
That was awesome. Thank you. I think I will also maybe include links to some of those online reports and articles
Sure.
in the follow-up email if anyone wants to check those out. That's very cool. So just to kind of go back to one of the slides that you showed, I think you combined it with root shield and with cease. And I think you maybe mentioned at the beginning of the presentation, like comparing like fungal versus bacterial based products. Did you find any trends there where maybe the chitosan was more effective at activation or
Right.
less?
Yeah. No, not necessarily. It did seem that the cease and the howler, I think we had better enhancement of
Mhm.
activity, and it may be because many bacillus bacteria are what we call chitinase producers. So they have the enzymes where they break down chitin and chitosan. And that's part of what makes them effective biocontrol agents for fungal pathogens is that they produce those enzymes. And
Right.
so they do this anyways, and we were just kind of stimulating them to do it more.
Yeah. Okay, interesting. Yeah. Um, and so we've got a few comments here. Matthew put in, there is a product, Easy Grow has a product with chitosan. So that is something that we're seeing for Easy Grow also provides a fertility supplement that is organic certified. So that's great. I did have a quick conversation with one of the people who works on the allowable substances list for organics in Canada a few weeks ago. And she did say that chitosan was on their radar. So hopefully they're in the process of making those decisions. And
I
I
think it
appreciate,
depends on where they get it from.
yeah, I appreciate that distinction because I didn't know that you could also get that from another source, which then would classify
Yeah,
that
I
as
think, yeah,
organic.
and that was where I think in the US it got stuck is that if you get it from crustacean shells, you need to, there's a chemical process to convert it to chitosan, and that doesn't happen in nature.
Yeah.
But if you get it, you can find chitosan in fungi, and then you don't have that conversion process. And so perhaps that would be considered naturally occurring.
Have you played with a fungal-based chitosan product?
I have not, because I think it's a relative newer
Okay.
idea.
Yeah. No, that's very interesting.
Evidence that it might actually work better,
Yeah. Hmm. Okay, that's cool.
but I
And
couldn't
then
get my hands on it.
no, not yet anyway.
Yeah.
Um, okay, let me see. Is the botrytis prevention on tomatoes only effective as a foliar spray, or do you think there would be benefit from soil application?
Yeah, so we've only looked at foliar spray, but I have seen reports in the scientific literature of soil application, so a drench application, for example, and I've seen reports of increased germination rate.
Oh, okay.
And yeah, so there's a sort of a plant, there's a biostimulant effect there. And then also potentially protection from soil-borne pathogens. So I know that that's been looked at. And so I know that it's been done with both um, products that are applied as a drench. I've also talked to a couple of growers. I talked to a tree fruit grower who swore by incorporating crab shells into the soil at planting. Uh, and that it just stimulated the microbial activity.
Interesting. I don't know if anyone on here has tried doing something like that, but that's, yeah, kind of go right to the source.
Yeah,
Um,
so no, in some cases, there's been a lot of research actually in Maine with potato growers incorporating chitosan into the soil. So the University of Maine has done that research with potatoes, and they've seen some good results. And I think a lot of it, and it's gotten a lot of press in Maine of taking like lobster, waste and utilizing this Maine produced waste for Maine potato growers, where they're actually just getting lobster shells and grinding them up into a powder and incorporating it into the soil.
Hmm.
They've been looking at suppression of potato diseases, both in the plants and the tubers.
And so I've also talked to our seafood development team at Perennia, because we have also had that issue with a lot of waste. So hopefully that's something that can kind of get that going, because it is a really cool
Yeah,
use of a waste product.
yeah.
Have you done any work with chitosan on strawberries?
We have not. Um, there there has been a lot of work on strawberry fruits for post-harvest shelf life.
Mm-hmm.
I'm sure there's work on foliar sprays that I can't remember off the top of my head. But in terms of a foliar spraying with flowers, I would imagine that you're probably in that similar realm where what was safe on petunia is likely safe on strawberry.
Yeah.
I think strawberries probably have a little bit thicker, especially as those leaves mature and they're a little bit more hairy than a petunia leaf. So petunia was probably on the spectrum of like really delicate.
Yeah.
And so what we could get away with without harming petunia is probably safe for many other things as well.
Yeah.
But yeah, we were able to go on tree fruits over 1%, whereas at 0.5% on petunia we saw phytotoxicity.
Yeah. Okay. So petunias kind of can be one of our maybe more vulnerable
Right. And
benchmarks.
if you wanted, I mean, many of the companies should, and that was part of what we communicated when we were working with Title Vision, was communicating that you need to have this information on your label about what's safe for different crops.
Yeah.
And so working with them, and also, whether you're applying it when the flowers are open and present or when they're not there yet.
Yeah.
So because the flowers are sensitive, and so it might vary based on whether the plant's flowering or not.
Mm-hmm. Okay. And I guess so then we have kind of along that same line of thinking, does the spray have to get directly on the blossoms to be efficacious?
Right. Yeah, we played around with that a little. We didn't get as far, so we did look at spraying the plants before flowering and then exposing the flowers to botrytis. So the flowers in themselves were never sprayed. And then comparing that to when spraying when the flowers were open. We saw better results when the flowers were sprayed directly
Mm.
than when they weren't, because essentially if you apply the chitosan before the flowers open, you're relying on a plant defense mechanism and not this direct interaction between chitosan on the surface inhibiting fungal growth.
Hmm.
So
Yeah.
it seems to be better when there's direct contact between the chitosan and the pathogen.
Interesting. Okay.
Yeah. I see a question about spray frequency.
Yeah.
We started getting into this, but for most all of the studies that we did in ornamentals, it was one application. And then we were exposing the plants to botrytis a day or two later. In tree fruits, I believe we were doing a 14-day spray interval.
Hmm.
So we were kind of just going out with the normal schedule for that orchard. So it was multiple applications. We've also done some orchard studies where we did 4 applications in a season. Because with tree fruits being outdoors as opposed to being in a greenhouse or a high tunnel, you're getting the rain that may wash it off.
Yeah.
And we did find that the addition of a spreader sticker made a big difference.
And
So we
apples.
1 year without a spreader sticker and we didn't get good results. And then the second year we added a spreader sticker and our efficacy was much better.
Okay. And I do have a question from our tree fruit specialist. Do you know if it has efficacy against black rot?
We did not test that. So
Okay.
we looked at fire blight, scab, the new one, Marcinina, which I forget what the common name of that one is. And then the post-harvest rots, blue mold, bitter rot, and we did some botrytis.
Okay. Awesome. Yeah, I'll share this with her afterwards. But yeah, she was like, I don't think she'd heard of it, so that could be pretty
Yeah,
easy.
pretty much everything we tested it against, except for Fireblight, we saw good results.
Awesome.
But Fireblight's another beast, so.
Yes, it is. And then, okay, so I have one other question, and it kind of goes back to one of your earlier slides where you're kind of defining like a biopesticide versus like kind of all the different groups of categories. I guess so is the idea that chitosan is not necessarily like the control product, but maybe more of a stimulant, or how is
Yes,
it classified there?
yeah, so you could probably do it either way, right? So when
Mhm.
companies are getting registration, they could go for the biopesticide route when they make disease control claims, because it
Mhm.
does have those properties. It does have the ability to suppress disease,
Yep.
but it also does seem, when applied in the soil, to have biostimulant properties.
Okay. All right.
But for the most part, I believe, at least for Armorzen and Tidal Vision, they were pursuing a crop protection label,
Yep.
claiming, so they're making the claims of disease protection.
And then when you were pre-exposing your products to chitosan, how did you do that?
Yeah, so what we did is we had a petri dish in the lab. So it was a petri dish with the nutrient media that was amended with chitosan, and we allowed the biocontrol agent to grow
Mm-hmm.
for a while on that, and then we transplanted it from that dish to another dish where it was exposed to botrytis,
Okay.
and that was the best. So it was something about the biological being able to grow in the presence of chitosan.
Hmm.
So it was activated, and then we saw enhanced reduction of the growth of the botrytis.
And have you
Versus
done
if you just put them both on the dish that had
right?
chitosan in it.
Yes. Okay. And then so I guess from like, have you taken that to the next step of like, I'm doing a tank, like I'm tank mixing these two together? I guess, do we see that same activation? Or is it like they need to kind of hang out for a while?
Yeah, I don't know, because we did the tank mixes, the simulated tank mixes, mostly just to see if they were compatible.
Compatible. Okay.
But we never then did like applied as a tank mix versus applied sequentially.
Hmm. Yep.
Because in many of the studies, what we did is we coated the flowers in chitosan, and then we sprayed the biological immediately after. So we
Yep.
didn't mix the two together. So I
Okay.
haven't compared mixing the two together versus two applications sequentially.
Mm-hmm.
But, in many cases, if you mix them together, and, by the time you actually prepare your mix and then
Yep.
you're applying it, it could be an hour, right,
Yep.
before they're sitting there together, and that might be enough of an activation.
Cool. Man, interesting
But I
stuff.
know some biocontrol companies are interested in chitosan for this reason.
Yeah. Yeah, that's really very cool. Okay, if anyone has any more questions. No? Awesome. Okay, thank you so much, Anissa. That was
Great.
amazing. So I'm
Thank you
just,
for having me.
yeah, no, I think that was really great. I will send out sort of a follow-up recording to everybody, and we will do the links that you presented,